So I'll start by welcoming Dr. Joseph Loscalzo to the podium. So he is a professor at the Harvard Medical School. He's very well known for his work in network science, among other topics. And I would like to ask him to start with the plenary presentations today. Thank you so much.
Thank you very much, Professor Pascalides. It's a delight to be here and to follow on the enthusiasm by the first set of speakers on this topic of AI and drug discovery. And so I'm going to have a little bit of something for everyone in the audience. We're going to talk a little bit about how the drug discovery process proceeds, what is a disease, how computational methods are changing strategies for drug discovery, and touch, of course, in the course of that on topics related to AI and drug discovery to set the stage for hopefully for the rest of the day. Here we are, my disclosures here. So drug discovery is a risky proposition, and I think everyone in the audience appreciates this statement, but it's risky in a variety of ways. And let me just highlight what some of the risk-related issues are. Here you see the annual yield of new drugs from 85 to 22. They really haven't changed very much over time, both for biologics and small molecules. The numbers are modest. There's a trend toward improvement, but not very much. The increasing cost of new drugs is another major challenge, and here you can see the number of drugs per $1 billion spent from 1950 through projected to 2030. So this is if you will, a losing proposition on its face. And more importantly, approved drugs lack precision, at least as we define them now. There are very high numbers needed to treat. Here you see an analysis by Shork that if you look at the top 10 grossing drugs that were approved by here in the US, you can see that the number of individuals who actually benefit from those drugs, highlighted in green, are truly the minority of all those treated with those drugs. This really speaks to the fact that while there has been a nominal p-value reached in a clinical trial, the benefits are modest, and part of that has to do with the economics of drug development, to be sure, but it also has to do with our failure to be more precise in identifying targets and pathways. So the next two slides highlight some of the key challenges in effective drug development as it has historically been pursued in the last 50 years. First, there's a persistent reliance on traditional paradigms, the sort of Ehrlichian magic bullet, single target, single drug for every disease. Absurd, given what we know about the complexity of chronic diseases. The NIH syndrome, not invented here. This is often found in big pharma. It stifles application of innovations or adoption of innovations developed elsewhere, or delays them. The over-reliance on conventional thinking about the genome, that everything is Mendelian at some level or other. The incomplete penetrance that we're alerted to really speaks to the fact that it is the genomic context within which a dominant mutation sits that really defines the extent to which the disease is manifest and how. Failure to consider off-target effects rigorously, except for those that are mandated by the FDA in adverse event screens. And the suboptimal screening sequence, identify the target, identify the drug, and then get a bunch of KOLs together to choose the disease target. That seems backward. There are other challenges that reflect institutional inertia, not the least of which is how we define disease. Most folks focus on disease definitions by ICD-10 codes, which include symptoms, syndromes, and diseases, and they're not always discrete or easily divisible, if you will. And there are convergent as well as divergent manifestations of disease about which we have to be alerted. What is the target? The FDA and EMA insists on identifying a target. Should there not be more than one target? Should we screen phenotypes instead of molecular targets? What's the organ? As we learn more about the physiome, we realize that very few organs operate in a complete vacuum. In fact, there is extraordinary crosstalk between organs that contribute to the extent to which a disease can be manifest. Target-based versus phenotype-based screens I'll touch on more in a moment. And then the inadequacy of the approach to off-target effects and adverse effects, as well as combination therapies. So the punchline for the rest of this talk is shown here. How do you de-risk contemporary drug development? Well, the first is, from our perspective, that we should look at interaction network-based approaches to target and pathway identification, as well as predicting adverse events through this mechanism and repurposing drugs through this mechanism. And of course, the use of network medicine, systems pharmacology, ML and AI approaches is paramount in achieving that kind of goal. So let's first touch on how drug targets are identified. I think it's easiest to think about this question or this issue from the perspective of whether one looks at target-based screening. Here's the target. I'm going to screen my 2 million compound proprietary library and find those that are most likely to bind and then set up a high throughput functional screen. Or phenotype-based screening, where we're not sure what the target is or even the pathway, but we do know that the phenotype is approachable through a less precise, mechanistically precise screen. And as it turns out, as you can see here in 2011, Swinney and Anthony showed that phenotype-based screening was more successful than target-based screening for first-in-class drugs. And at that time, we knew far less about the complexities of the network interactions responsible for phenotype, but it really highlights the fact that we honestly don't always know what we're doing. We know where we want to go, but we don't really know the underpinnings. So here's a conventional reductionist approach to drug target identification. We have an input and an output. We have from GWAS screens or genetic studies or perturbation screens or literature curation, as shown in the red dot, a target. A drug is tested against that target, and we measure the consequences of affecting that target on the conversion of input to output. Pretty straightforward. But notice the other dots and edges that are shown there in that dashed space. Those reflect the complex context within which that protein, that red dot, is operating. And that complexity manifests as other emergent properties which can be crucial to defining the effect of a drug or the lack of effect of a drug and the manifestations of disease in patients who carry that mutation. There is allostery, well-known biochemical phenomena, where effects at a site separate from the critical active site of an enzyme or binding site of a receptor modulate that protein's function through another mediator, another allosteric activator or suppressor. Oscillations can occur, even in a linear pathway, as shown in this study by Yang in 2008, which is clearly an emergent property and reflects the balance of rate constants around the different enzymes responsible for the pathway. There are many other types of emergent properties, but these are two very obvious ones that one needs to keep in mind as you think about this more integrated approach to drug target identification. And this approach that myself and my colleague Laszlo Barabasi began to think about and published first as a concept piece in 2011 is represented in the following way. That if you take the universe of interactions among all the proteins in a cell, within that universe of protein-protein interactions, there are subnetworks, as shown in the pale oval there, which reflect those interactions that are crucial to the development of a disease. And that hypothesis was tested and proven in a subsequent paper here published in 2015, in which at the time we had 13,500 proteins that interacted through 140,000 different edges or associations. A very complex, dense network within which we were able to identify these modules that are disease-specific, some of which overlap, as shown here schematically with rheumatoid arthritis and multiple sclerosis, both of which are autoimmune diseases. You'd expect some overlap. You can build a disease module on the basis of some knowledge of a data set that associates with a phenotype or a disease. And here's an example for cerebrovascular, ischemic cerebrovascular disease. You identify the phenotype of interest, and you ascertain the disease network components or seed proteins from the curated literature or from an unbiased screen. You map the seeds to the unbiased interactome and then identify connector proteins that link the seed proteins, usually not more than one edge apart, which then statistically can be proven to be a distinct largest connected component that subtends this disease module. What's the benefit of doing this kind of exercise? Well, the benefit is that the interactome provides the missing links among the disease-associated proteins in the disease module that give you much more insight about mechanism and potential drug targets. More recently, we've defined disease modules using a quantum-inspired approach. This was just published this year, where we use a quantum walk across the PPI to identify that collection of proteins which most directly associates with a specific disease phenotype. A much more efficient usage of this is a time-dependent continuous time quantum walk approach that we're now beginning to apply more broadly. In work that Fei-Xiong Chung did when he was a fellow, he demonstrated that there's an association between this network strategy and conventional views of genetic mutations and their association with disease. And what he found in this first paper published in Nature Genetics was that mutations that are associated with disease are 6 to 8-fold more likely to be associated with or occur in the interface between two protein binding partners than the exofacial sites. And that's true for germline mutations as well as somatic mutations. And in a lovely paper he and his colleagues put together, just published this month in Nature Biotechnology, using a deep learning inspired strategy called Pioneer, he was able to predict the binding interfaces for all human proteins and 7 other species. So another important point to make about this network strategy for drug development is to think about the fact that each of us has our own networks. Every disease is unique, even the most common diseases. That's an important punchline to keep in mind. And here's an example of that point. Here's a big, thick heart. Those of you who don't look at hearts won't recognize how thick the walls of the left ventricle are, that portion to the right of the slide. This is a heart from a patient who has hypertrophic cardiomyopathy, well known, fairly common among genetic causes for cardiovascular disease in adults. Cardiovascular disease, a cause of heart failure and sudden death. Mutations have been identified in more than 11 sarcomeric proteins, the proteins that comprise the contractile apparatus of the heart muscle. And the disease is highly variably penetrant. As it turns out, only about 1/3 of patients have these sarcomeric mutations. So this represents a convergent phenotype. Many other causes of this phenotype exist, not just conventional HCM mutations. And using a network-based strategy, a topological network-based strategy, Rusheng Wang and Brad Marin and our group demonstrated in samples taken from 18 patients with HCM who were undergoing septal reduction surgery so that we could obtain the messenger RNA from their heart tissue compared to normal heart donors that were going to be transplanted into patients. You can see that these clouds, these fuzzy balls that you can see for each patient, are quite different even from this altitude. And this allows us then to subclassify patients with HCM into different reticulotypes, as we call it, after the Latin term for network. And within those reticulotypes, you can see different disease drivers, which begins to allow us to become much more precise in identifying pathways that should be targeted for patients with this disease. So molecular interaction networks inform genetics and facilitate genetically derived drug target identification, and tissue-specific gene expression informs interaction networks and can, at least theoretically, guide precision therapeutics. So now let's just touch a little bit on network-based drug repurposing. First of all, and some of you may be surprised at this slide, that drugs have many targets, despite the fact that the industry prides itself on being rather precise. Remember that it's precise to the extent that a single target is explored, in general, as the cause of a phenotype and screened with respect to proprietary libraries. But as this analysis by Chartier showed, using a proteomic bioinformatic analysis, that the average number of times that a similar binding site structurally appears in the human proteome for drugs that have been approved, whose binding sites are known, is 25. And notice that it's a very long-tailed distribution. So it is the rarity for a drug to be truly uniquely interacting with a single target. What does this mean? Well, the response is, well, this accounts for off-target effects. Well, that's true. But it's also an opportunity to consider alternate uses for drugs that have been approved if you knew what those other targets were. So for that reason, we set out on an effort with Felice Lightstone at the Lawrence Livermore Laboratory, where they have a near exascale computing system, the Sierra system. And what we did over a period of about 18 to 20 months was screen over 28,000 models for 14,000 human proteins. Either their crystal structures were known or later on AlphaFold derived structures were used against a 2.5 million compound library, and created a matrix of drugs and targets. And now you can basically walk down the row or column of that matrix of your interest and identify a potential drug for an interesting target that you think it may be relevant to a disease module you've identified. So that really speaks to the fact that this network-based drug repurposing proximity hypothesis shown on the right argues that if there is a target not within it that if there is a target for a drug that is not within the disease module for the disease against which the drug was developed, that might be an alternate strategy for developing the drug for a new purpose. And we have done so in a variety of, by a variety of examples, one of the more recent ones of which had to do with SARS-CoV-2. In this case, we identified the drug target disease module as well as the COVID-19 disease module, a so-called foodome, and this represents the human proteins that interact with the 30-odd SARS-CoV-2 proteins. And once we had the foodome identified, we used a proximity strategy, a diffusion strategy, and a network-based AI strategy that Marinka Zintnik was really responsible for. And using these methods, we prioritized FDA-approved drugs. We used the broad pool of FDA-approved drugs, about 8,500 or so, and identified, as you can see, each of these three methods were complementary with respect to ranking information. The Kendall-T rank correlation of the rankings was provided by each of several pipelines within each of the methods. So using these correlations across these three methods, one could create a rank order. We tested those drugs in here at the National Emerging Infectious Diseases Laboratory in Davies' lab, and Justin Patton did this analysis. So we identified those drugs that had virucidal activity but not host cell toxicity. And here you can see that the closer the drug is, that is, the lower the Z-score, the more likely it was to have an effect that was appropriate for the virus, for virucidal benefit. And here you can see an example on the right in particular in which we expressed the human ACE2 protein in mice and demonstrated that with obatoclax, one of the drugs that came out of the screen, which is typically used for certain kinds of lymphoma, that we could greatly reduce viral load as well, not shown here as survival. Now, there are many commonly assumed flawed assumptions in experimental validation of drug candidates. And I'm going to list four of them here. This may lead to some discussion at some point today. First, intracellular diffusion of small molecules follows Einstein's laws. That's not true. It's anomalous with a time-dependent diffusion coefficient, as Vlad Elgard and our group showed a couple of years ago. Solution phase binding constants and kinetic constants reflect intracellular constants. They do not. They're often 1 to 2 orders of magnitude different from solution phase constants. Intracellular concentrations in proteins and metabolites are sufficiently well known and reflect solution phase constraints. Neither of those is true. The intracellular concentrations of most proteins are not known, and many metabolites' intracellular concentrations are not known. This, from the standpoint of building models, AI-driven or otherwise, these are absolutely crucial bits of data which we do not have, and being able to sort them out would be critical to being much more precise in our predictive accuracy. The predominant structure, and this is important, especially for some of the discussions we'll have today, the lowest energy state of a protein can best be approximated by the structure that's crystallizable. This is not necessarily true. In fact, cryo-EM images prove otherwise. You can have a whole ensemble of structures for many proteins, many of which no doubt are in a relatively low energy state. So which of these is responsible for the functional consequence that you're trying to in some way modify with a drug? So we need alternate strategies to predict associations. And Giulia Menichetti, who has worked closely with us over the last few years, Giulia Menichetti developed an algorithm called AI Bind, which importantly utilizes both information available through conventional binding strategies, energy minima, pocketome analysis, but also that's coupled with many other features that AI can incorporate with respect to the physical chemical elements of the drug as well as other factors that contribute to much more precise and more predictably accurate approaches. So in the last 2 minutes, let me just talk a little bit about another approach that one can use to identifying drugs. And this has to do with our efforts to address this notion of food as medicine, very hot topic in nutritional science. We were just awarded a major grant from the AHA to pursue this project. And the point here is that you would think, based on all the epidemiology that's been conducted over the last 60 years, we know a lot about the small molecules in the food supply. Well, as it turns out, epidemiologists have only focused on about 188 of these small molecules or nutrients. From our analysis, and this is Julia's work and her team's work, of the available data sets that she explored worldwide, the foodome database that she's now created has over now 140,000 compounds, 93,000 of which have been detected and 47,000 of which have been inferred. Take garlic, for example. There's 69 of the known nutrients in garlic that the USDA recognizes, but 6,800 that are not considered. So we talked about this unmapped chemical complexity in our diet, or the dark matter of nutrition, as a potential source for drugs. And in work that Hao Shu's done, that's now in review, what he first has done is basically used a UMAP and former pre-trained molecular structure AI model to explore the universe of classes of small molecules in food. And here you can see a depiction of that two-dimensional depiction of those classes. On the left in blue are a whole host of lipophilic molecules. On the right, there are more polar molecules. And if you can read from back at the back of the room, you can see what some of the subclasses are. But I show you this as a backdrop to this slide, which is also in the paper that's under review, where we compare this nutrient dark matter to drug bank databases. And notice how much smaller the drug bank information is, more condensed. Obviously, there are fewer entries in the drug bank, but still, they're all very well clustered, far away from the lipophilic domains that you saw in the last slide. But also, probably because of Me Too chemistry, once you find a winner, everybody works around the winner in its pathway. That's what happens here. So we have an opportunity to explore this foodome, if you will, as a source of drugs. It has evolved in parallel with humankind. Therefore, we know a lot about the safety of those compounds because they've been in our living organisms for millions of years. And our goal as part of this effort is to use both high-throughput strategies, AI-based strategies, to identify compounds that might map to the targets in the interactome. Half of the 140,000 have already been screened through the Livermore analysis that I mentioned to you. The other half are being screened. And therefore, we can develop a platform for identifying potential new drug structures derived from food molecules, but also identify diets that might provide the kind of healthful benefits that we all seek, especially in food deserts and especially for folks who are more socioeconomically limited. So drug development is risky. Evolving AI-based approaches filtered through the lens of molecular interaction networks, coupled with rational experimental observations, I think holds promise for efficient and effective drug development. And just give a pitch here for a symposium that was held, that was supported by the Menarini Foundation in Milan last year on AI and network medicine as a path to precision medicine. I acknowledge those folks who did the work that I shared with you, and I thank you for your attention.
Thank you very much. We have time. We can take one or two questions, just one or two questions, and then obviously Dr. Loscalzo will be here in the break for additional questions. So any questions from the audience? And we have a microphone, so if you are interested in asking a question, the microphone will come to you. Let me ask one question. Your talk, I think part of the role of the AI and of the data science methods was in essentially screening a number of different compounds and also in sort of discovering the network structure that is important. Where do you see the role of generative AI methods in terms of designing rather than screening known compound databases? Any comments on that front?
I think one of the things that falls out of that Howe-Shu analysis is that using QSAR, or more conventional structure-activity relationship-based drug design for expanding the pool of potential drugs, has its limits, as anyone in pharma knows. So I think at that level, there are features which may not be apparent based on the conventional physical, chemical, or organic chemical parameters that we would follow in drug development that generative AI may guide us to. Interestingly, I've given Howe the task of identifying compounds in the pool from the food database that we might test against known targets with well-defined drugs useful as inhibitors, like HMG coreductase or angiotensin II receptors. And some of the compounds that were generated or were identified in that pool were obvious, but then others were completely unobvious. They bore no necessary physical, chemical, or structural relationship to the families of drugs that had been developed through conventional QSAR or SAR over the years. So I can't tell you whether that works or not yet. That's ongoing work, but I think it's an interesting possibility.
All right, thank you very much. Diane has a question.
Oh, yes.
When you were comparing the food to the known drugs, I'm just wondering, is part of the reason that the drugs are all in one space because of their physicochemical properties? And do you think that the rest of the space would be able to get into cells and act as a drug?
Right, so very important issue. So I think there are at least 3 different reasons for that clustering. One is the Me Too approach that I mentioned. Another is just the feasible use of certain compounds. Are they sufficiently soluble? Are they transported across membranes? How are they metabolized? Some may have toxicities as well, which in unique situations. I think all of those are possible. I will say, though, and I didn't show you this, but if you look at the polyphenols, some of which are drugs, if you look at polyphenols, they extend beyond that conventionally, that tight cluster in the middle of pharma-developed compounds, in part because they're more complex chemically, in part because they're redox active and you may not always have the species you think you have when you give it to a person. So I think that it's a combination of all of those features that account for it.
All right, let's thank Dr. Loscalzo.